Before starting any of your processes, you need to ensure that you've undertaken hand decontamination through hand washing, and that you're wearing all the required personal protective equipment that is appropriate for the task you're about to undertake. These will be listed in your standard operating procedures and will include items like lab coats, gloves, goggles, and masks as needed. Once you've finished with the hand wash and put on your PPE, you'll need to prepare your work areas. This includes taking out any of the consumables that will need to be brought to room temperature, as well as cleaning down your workspace. You'll also need to do things like prepare disc pots that contain 10% bleach. Your workspace must be cleaned at the start and end of every day, as well as in between changes of procedure when you'll be working with different specimen types. The same process will also need to be undertaken if there are any spillages. For fluid spillages, you'll need to follow local guidelines, as there may be additional steps that you're required to undertake. Preparing and cleaning down of your workbench includes not just cleaning down the bench space itself, but any of the equipment you're likely to use, including racks and pipettes. We're now going to cover how you store bacterial colonies in glycerol stocks. There are multiple points across these processes where you may need to store bacteria. You will always be storing a sweep in order to try to store a representative sample. You'll need to store 2 vials from the primary plate. The labelling for the stock should include organism/stage stored, specimen ID, visit number, date, and vial number. You may also need to affix these details either with a barcoded label or manually with hand labelling. You'll also need to store a minimum of 2 vials from any secondary plates, more if different morphologies are present, and 1 vial per isolate from an antimicrobial resistant plate. If available, you will store Streptococcus pneumoniae isolates in the blue capped vials and E. coli isolates in the red capped vials. Primary sweeps can be stored in any coloured vial but need to be identified as such. The coloured vials will allow easier differentiation if you are adding sheep's blood to glycerol to better support retrieval of Streptococcus pneumoniae. It's key, therefore, to ensure the right organisms are stored in the correct vial. If colour-coded vials are not available, then labelling with the correct organism type is crucial. Now on to the process itself. Have your barcoded labels that you will affix to match the specimen identifiers, or have your pre-labelled tubes present. For each bacterial sweep that needs storing, take a 10 microlitre disposable loop or flamed metal loop under aseptic conditions and take a sweep from the plate and open the screw cap of the storage vial. Inoculate the cryopreservative fluid by twisting the loop in the fluid multiple times, making sure not to spill the fluid. If this is your primary culture plate, ensure that you've captured all the different bacterial colony morphologies and the species within the sweep, whether they are target species or not. This includes opticum resistant colonies and non-blue violet colonies on chromogenic media. Once you've inoculated the cryo vial, close the lid and ensure you mix by inverting 4 to 5 times. Do not vortex. Vials can now be frozen at -80 degrees. Once you've prepared the vials, you need to record where the vials have been stored in the logbook, including the position in the cryobox number, as well as the position in the freezer. You will then need to clean down your bench space appropriately.